Okay. Hello, everybody. All right, 4 lectures left. We're in the home stretch. I just want to, for those Colin gave a great talk on the insula, so the other 3 GSIs are each going to give a 10-minute talk on something they think is salient to this course that they're going to present. So today's going to be Mano, who's going to talk, and I'll introduce her after I finish the section on hemispatial neglect. And then the last 3 parts of the last 3 lectures, really, the aphasia and disconnection syndromes are all part of the same thing, hemispheric organization of processing and particularly language. And then we'll close the course with a pretty nice little talk on visual agnosias, which are perceptual distortions. So hopefully by this time in the course, you've got a lot of infrastructure to start to begin to figure out what's going on with the patient, what you might want to order as a test, what may or may not support or not support what you're doing. So with that said, this thing should be in here. Oh, it is. It's not working. Lovely. All right. Huh? Almost. I thought I had everything working. One little flaw. We finished the course, the last lecture, talking about this global local analysis property, which is this incredibly rapid ability of people to change the reference frame to seeing the global aspect versus the detailed local aspect summit and so on. Easily shown with patients with either right or left hemisphere lesions copying these classic Navone figures. And you show them the stimulus, and you can see the stimulus quite nicely. I just figured out why the advance wasn't working. I did not have the advance on. So an operator, I was so excited. I was doing well. So these are the stimuli again, and it's really a classic finding. You see it in the patients right at the bedside. You don't need some big, fancy analysis. It's quite obvious. Right hemisphere, particularly right temporal parietal damage, basically loses the global aspect of the scene, gets the details, and the vice versa with patients with lesions in the left temporal parietal. Really interesting observation. You're doing it so fast you don't even know. Your attention window is being shifted back and forth between your right hemisphere at a subconscious perceptual level, which in general is under 300 milliseconds. And most of this attention switching is done by roughly 200 milliseconds based on electrophysiological data. So the chronic syndrome is a little bit different than aphasia, which you'll see starting at the end of today and then going through Wednesday. People with severe aphasic disorders often are quite symptomatic at 2 months, 6 months, 1 month, 5 years, even 10 years. They can have permanent problems. Patients with hemispatial neglect often show substantial improvement. The denial of illness, the anosognosia, is usually gone in a few weeks, and then they slowly improve. And often by a year, it's hard to pick up without more sophisticated testing, maybe extinction and attention deficit, where they still may remain with a left hemiparesis, for instance. So I want to emphasize again this idea that this problem with neglect is a deficit in the internal representation of space. It's how you see some picture you want to look at, either consciously in this room, and that part doesn't make consciousness, or you engender a picture in your mind, as I showed you with that Piazza di Milan, where people knew everything was there. Depending on their viewpoint, they could access it. There's a series, maybe 12 or 13 in the literature, of famous painters who've gotten hemispatial neglect and painted themselves acutely, where they leave off half their body, and then acute, and at 2 weeks some returns, and by 6 months they paint their whole face. So they've had a slow return of the ability to access their internal representation of how they look, not just how the external world looks. So the chronic syndrome, you can see the parietal lobe deficits, like elementary things, graphesthesia. You can't read a number on your hand. You can't perceive things. These are high-level sensory, but not attention. Oftentimes, you'll see extinction And you can see it in all modalities, but it's usually much worse in visual, greater than somatosensory, and then auditory. Now, real interesting phenomena, again, getting at this critical window of conscious perception, is if I do this with a patient with neglect, and the patient has a right hemisphere lesion, and I flash both fingers. If I flash this finger, they say left. If I flash this finger, they say right, right, left, left. They extinguish. If, however, I make the two stimuli asynchronous by about 300 milliseconds to 400 milliseconds, like this, they see both stimuli. I don't know what it means, but it's pretty interesting. And I'll show you an example of it. So this is a gentleman he's about 3 months, maybe 2 months, roughly post his stroke in the right temporal parietal area.
Put a cross through every line that you see there.
So this is the opposite line bisection test, which we've already gone over, a very sensitive measure for hemispatial neglect. Do
you think you're having trouble seeing things on the left side?
Oh, yes. I have to, when I read, I have to make sure I go all the way over to the left side to start the line. A flower.
Flower in a pot.
Internal representation of a flower. Half of it is not drawn. Classic.
I didn't do that very, very well.
Okay. Great. And
bedside.
What letter do you see there?
S.
S.
He sees the details. He doesn't see the global.
What letter do you see?
Right at the bedside.
Okay. Ready, sir?
Yep. What do you want me to do?
Left click, right click, or both.
Oh,
Ready?
Okay. Right click. Left click.
Ready?
Yep. Right click.
Extinction.
Right click. Left click. Left click. That was right click.
Okay. Okay.
So he's got the hardest to see usually is auditory extinction.
Right click.
Okay. Temporal synchrony.
Right click.
I still can't get it with the figure.
That was both.
Both.
Okay.
When I got it about half a second
Right.
apart, he noticed both.
Right.
Extinguish.
Right. Left. Right. Right. I think it was both, but
Not sure. Okay.
Left. Right. Left. Right. 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 Left.
Classic, right?
Right.
Extinction. Right. This is just
Left.
textbook.
Right.
All the clinical, and he's over the really severe period. He knows he's got a problem. He doesn't have anosognosia. He's still having problems with the left side of space. He's on his way to some recovery. Yeah. A couple ones where he wasn't sure. There's a really interesting, there's been a couple studies where people have taken these patients and wired them up and recorded their EEG and recorded the visual evoked response on neglected versus non-neglected versus not sure trials. And when they really have prominent neglect, there's a severe suppression of the visual activity in the hemisphere of the lesion. And when they almost get it, or maybe even get it, they show increasing activity. So there's a physiological gradient of how much your attention network is suppressed on a trial-by-trial basis. But that's a really, that's exactly what was going on with him. He wasn't sure. And that's the reason. I think he's got incomplete information in his visual cortex. Right side is most common by far. The network is pretty broad. The biggies are temporal parietal cortex and frontal eye field. But, if you get a good-sized thalamic lesion, you're going to get neglect. I've seen it with, I doubled the perseveration. Basal ganglia lesions and even cingulate lesions. These are much less common. These are extremely common. 90% of cases have some degree of right temporal parietal damage, just like 90% of cases, as you'll see with Wernicke's aphasia, at least have some degree of left temporal parietal damage. The most common cause, not surprisingly, is middle cerebral artery infarct, because that's the vascular distribution, right? That's the middle cerebral. That's the posterior cerebral. This is the anterior cerebral. It's right in that distribution. And again, it's not a sensory problem. We've already gone over that when we showed, basically the patient with hemianopsia actually will search the visual scene and find all the objects. Now, the anatomy basically is based on these very strong connections. This is monkey anatomy. The frontal eye field in the monkey. The frontal eye field is a key output for your attention network. If I stimulate it, your eyes and body go left, right. If I stimulate the left side, your eyes and body go right. Very important for allocation of attention to the world. Covert allocation. Same thing in humans. This is the key network. Area 8, which is frontal eye field, and this temporal parietal junction. Again, this is just more a little bit, just another anatomy slide, laying it out in a little more detail on an MR scan. There's your temporal parietal junction. The Bruntal lesions are there. But you can also have it from isolated frontal eye field. The most severe cases get both, which you can get with the middle cerebral artery infarction. Just like the most severe cases of a complete occlusion of your left carotid or left middle cerebral, as you'll see on Wednesday, is a severe aphasia where you both can't comprehend and you can't output. And as I mentioned, occasional cases from the basal ganglia and the pulvinar, particularly pulvinar hemorrhages, hypertensive hemorrhages in the thalamus. If they're big enough, there's a massive projection from the pulvinar, which is the back of your thalamus, to the temporal parietal junction. So if you actually take a patient who's got hemispatial neglect and they've got a big pulvinar lesion and you put them in a PET scanner, not so surprisingly, you see hypometabolism in their cortex because their thalamic projections are disrupted, and that's what's going on. You're knocking out this thalamocortical network. Again, this idea that almost everything we do has some property of a network function. Again, here's some of my cases from a long time ago. This is how we used to kind of show lesions in old papers. We didn't have CT even. We would just overlay them on a brain. Here's an actual 3D of a patient with a lesion in the temporal parietal junction. And that's this is a pretty old MRI scan, because if you look carefully, the way you can tell it's old, first it's grainy, and the detail now is it's much higher in terms of the pixel resolution. But grainy could be because the patient's moving. If you look carefully, see how the slices are lined up? They're not perfectly lined up. That tells you it's a really old technology. Now they get lined up really quite nicely. Here's the worst cause, which is severe neglect. And again, this is the right hemisphere. So basically, what you see here is a big chronic middle cerebral artery infarct. It spares the anterior cerebral. It spares the posterior cerebral, the hippocampus, and the visual areas. But it takes out a large part of your hemisphere, both frontal eye field, which sits right about there, and temporal parietal junction, which is down about 2 centimeters. Now, you can get other weird causes. This is a very sad case. When I was first at Berkeley in '85, there was somebody who was breaking into, burglarizing people's houses and basically beating up people. And he broke into a house on Virginia, which is really it's only like, 4 blocks from here, 3 blocks from here. And basically there was a nurse there, and the guy attacked the nurse with a hammer. And that was his preferred weapon. And you can see here he actually hit her in the skull there. And that's how the skull should be. There's a depressed skull fracture. That's where the hammer hit. You can see a contusion. See that white signal? That's a contusion actually in her brain, and she had hemispatial neglect when she presented to Highland Hospital. Luckily, she recovered. The guy actually got caught. He was a city councilman. And there was yeah, say that what again? Yeah. Say what? Yeah, exactly. And there was a witness against him. I'm going to get this right. It'll be out there forever, but I'm pretty sure I know he killed the witness, and I think he beheaded him. And he finally got caught, and I hope he's still in prison. In any event. So it doesn't have to always be stroke. It's mostly stroke. I've seen patients with gliomas and tumors in this area and other forms of trauma, not just hammer attacks, producing the neglect syndrome. Here again, just to show you, you can get stroke from can get neglect. This is a big white lesion in the thalamus on a CT scan. There's the bone, And you can see that's a hemorrhage in the pulvinar. And again, if I put the person in the PET scan, this whole part of the brain would be hypometabolic because it was losing all its projections. And these patients can certainly have neglect. Again, once more time, the system's nature. So the theory is that there's a hemisphere, I'll just give you the theory. There's a hemispheric asymmetry for attention. There is some support for this because if you do quantitative MRIs of humans, you will notice that the right frontal lobe is slightly bigger than the left. In terms of language, your left parietal, temporal parietal occipital area is actually a little bigger than the right. So there's some anatomical asymmetry which you don't see in other syndromes other than neglect and aphasia. So the idea is, and I've already talked about it's a deficit in the internal representation of space. The idea is, why did it become asymmetric? The classic line, and I don't know if it's true, they may have just developed in parallel for some other reason, is as language developed and took over the left hemisphere, you had a kind of push over to the right hemisphere of some of your attention capacity. Not all of it, not as strongly lateralized as language, but it really was a way to use other resources in the brain. So let's just see a little bit evidence for that. So this is how the idea goes. The left hemisphere allocates attention predominantly only to the right hemispace because it doesn't have enough resources to do much more than that, since so many of its processing resources are being dedicated to what we do like right now, which is language. That's the idea. Conversely, the right side of the brain has evolved, particularly with some focus on increasing size of the right hemisphere slightly. The right side of the brain can allocate attention to the left hemispace, just like the left brain can do right, so it can do left hemispace, but it's also evolved to allocate attention to the ipsilateral hemispace, right? So it has its attention capacity, whereas the left brain can only do the right side of this room on attention. The right brain can do the whole room. Okay? That's the idea. So it's like this. The right hemisphere can do both fields. The left hemisphere can only do the right field. So you can see what the result is pretty simply if you lesion it. If you lesion the right brain here, the left brain will still take care of the right hemispace, but there's no way to do this hemispace because the right brain is gone. So the left neglects, since the left hemisphere cannot allocate attention to the left hemisphere. It's a very simple idea, but it does fit very nicely with the actual behavior that you see routinely. Now, when you get a left-sided lesion, which are just as common as right-sided lesions, the vascular tree is, pretty stupid in the sense it gets disease and atherosclerosis and emboli to either one. I shouldn't say stupid. It's just, it's plumbing. If you get a left brain lesion, you can see the logic again. There's no or minimal neglect since the right hemisphere can allocate attention to both hemispaces, and that fits clinically with what you see. So it's a nice kind of clinical theory connection. A very important syndrome to be aware of. All right, so now I'm going to turn it over to Mano. Mano is a second-year grad student. She's had experience as an undergrad working in labs where she's done both functional imaging and EEG research. Her research now is in the lab of Sherry Johnson. It's a clinical psychology lab. Mano is interested in general and bipolar disease and other major psychiatric disorders, and that's what she's going to be focusing her thesis work on with a lot of interest in reward learning. So I'm going to turn it over to Mano, and she's going to give you a talk on what she would like to add to the course.
All right.
That's it.
How long
That's
do you want me to go with this? 10 minutes?
10 minutes would be perfect.
Should I wear the microphone?
You should wear the microphone. You have to wear the microphone, or else you don't get streamed. That'd be terrible.
Oh, so you don't do notes?
Huh?
That's okay.
I've never used. Should I use notes?
That's all right.
I might be more
It's gonna
coherent
be
if I had.
improv.
You do notes? For your?
Yeah, usually. Okay. Cool.
All right, so you're good there, and then this is the slide advance, that one, and that's the.
All right.
Okay.
So we've talked about reward and reward processing a little bit in this class already, kind of peripherally. So we talked about how reward is really important when it comes to processing emotion, and also about like people who have damage to the prefrontal cortex show problems with drive and motivation. And if you think about it, we wouldn't really be able to study much of cognitive or behavioral functions in general if motivation wasn't there in the first place. So I think reward is pretty important. So I'm going to talk a little bit about how we process reward, both in terms of what that looks like in the brain and also how we measure it with behavior. So this is showing kind of a basic image of the brain's reward circuit. So dopamine is produced in the ventral tegmental area, and you see projections going to the nucleus accumbens and to areas in the prefrontal cortex. And this is generally what's talked about when you talk about the reward circuit in the brain. Again, kind of a simplified image, but just to give you a basic idea. And then reward processing itself is kind of a broad concept, and you can really separate it into at least these 4 different subcomponents. So you can talk about reward in terms of liking, wanting, learning, and also willingness to work. And I'm going to talk mostly about the first 3, and I think probably won't have time for the last one, but just give you a sense of where we're going. So when you say liking, what this really means is generally response, basically hedonic response to some kind of pleasurable stimuli. So one way this is measured commonly kind of across species is with this sweet taste test. And what you see is that the observable responses in both, I think those are mice and humans, is very much the same for liking versus disliking. So again, this is this momentary response to pleasure, and you see for liking, these kind of tongue protrusions, licking behaviors, and for disliking, in response to bitter taste, you see mouth gaping, and I know it looks kind of similar in the pictures there, but the idea is that there's these observable differences. So liking looks very similar across species. And so what does this look like in the brain? We've talked about how the orbitofrontal cortex is important for coding valence, or basically amount of pleasure of a certain stimulus. So on the top pane there, you see in both the images of the brain, the orbitofrontal cortex area is circled, and the top pane is showing response to pleasant smell, and the bottom pane response to pleasant taste. And the interesting thing here is actually that the degree of signal you see, of bold signal, correlates with the actual subjective pleasantness rating, both for taste and for smell. So again, orbitofrontal cortex involved here in this immediate response to pleasure. Orbitofrontal cortex is not the only region implicated here. You also have other kind of hedonic hotspots like the nucleus accumbens, which we'll talk about in a moment. So another way to look at how people experience pleasure is in terms of anticipation of reward. So if you think about like planning for a vacation, can be kind of just as exciting as being on the vacation itself. This anticipation phase is also highly rewarding. So in the case of anticipating pleasure, one way that this is measured in humans is with, for example, the monetary incentive delay task you see here on the right. And in this, people learn to associate a cue with a certain amount of reward value. And what you see in the image on the left is nucleus accumbens activity, bold signal, in response to the reward cue, but not the reward itself. So what this is showing is that people are responsive not just to the actual reward, but also to signals that are suggesting reward is about to come. And in particular, again, you see the degree of signal increases with the amount of reward that the cue is signaling. And you can see on the graph, too, that this signal is sensitive to increases going up from 0, but it's not sensitive to kind of the amounts when you're losing money. Again, suggesting this is really specific to reward and not necessarily loss. So I won't really focus on this slide too long, but I want wanted to mention that in terms of self-report, researchers are also looking to be able to measure how people kind of experience these things differently. And when Devin talks about mood disorders and reward next week, you'll see that in some cases people have intact subjective liking but not intact wanting for reward. So these are really separable constructs that we can measure, again, with behavior and also with self-report. So what about learning? So this is one kind of really interesting study that based on the idea that I think a long time ago people had this idea that dopamine really codes pleasure, right? It's the pleasure neurotransmitter. And in this study they showed that that's not quite exactly the case. So this is a monkey study. And in this study they were giving monkeys juice, and then they taught them to associate a tone with the juice, through classical conditioning. And then they used single neuron recordings to look at amount of dopamine firing in response to both the cue and the reward. So on the top pane there, that's when they're just receiving the juice. They haven't learned any associations yet. And you see an increase in dopamine firing right at the point of receiving the juice, right? This liking response. Okay. But the interesting thing is in the second panel, once they've learned to associate the tone with the juice, you see an increase in dopamine firing in response to the tone, but no increase in response to the juice. So if dopamine really was just coding pleasure, you'd expect to see also increased firing in response to the juice, right? And then on the bottom pane, what you see is, again, the monkeys are showing this increased dopamine firing response to the tone when they've learned to pair it with the juice reward. But they don't actually receive the juice reward in this particular case, and you see a decrease in baseline dopamine firing at the point of reward where they would expect to have it. So what this is really showing is that dopamine is implicated in its prediction error signaling. So kind of showing differences in expectation about reward rather than just coding for the amount of pleasure and again, this is monkeys, but we've also been able to look at prediction error signaling in humans with bold signal and other measurements. So again, this in humans is the way that we might measure this. So with probabilistic reward tasks where people basically learn to perform certain behaviors based on the feedback that they're getting. So this is more operant conditioning. And since we don't usually do single neuron recordings in humans, one way that dopamine levels are manipulated are through pharmacological manipulations. So if you increase levels of midbrain dopamine in people, they'll actually begin to behave in ways associated with more reward sensitivity. So they'll start to get more, they'll be more sensitive to feedback, and they'll start making choices more associated with rewards than they would if they didn't have that increased level of dopamine. You can see the opposite pattern. If you give a drug that decreases levels of midbrain dopamine, like haloperidol, people are less sensitive to this reward feedback, and they're not behaving in a way that would suggest that they're going for the reward as much. So in a sense, you have increased or enhanced reward prediction errors when you increase levels of midbrain dopamine. You have a question? Yes. So this is the way that they're measuring this sensitivity. So with this kind of task, you basically have to try out different options of choices and figure out through that which symbols are more likely to be associated with reward. So increased choice probability towards gains is just the way that they measure. People are more likely to make choices that have previously been associated with reward. All right, and I think I'm almost out of time here, right? But another fourth thing I just wanted to mention was, so in some studies with animals, they found that, when rats press the lever to get a reward, they found that the amount of dopamine signaling going on was more associated with how much they pressed the lever than it was with the actual quantity of reward that they consumed, which might suggest that dopamine plays an important role, not just in, the actual anticipation of reward, but specifically in a willingness to increase effort towards that reward. So I won't talk too much about this, but if you have questions, you should talk to Nick, because he's using a task that, well, I guess we are too, but he's the effort task man. So. Okay. So that's pretty much it. I mainly wanted to give people a sense that there's different ways of measuring reward processing. It's separable into these different subcomponents, and you can see differences in these areas, both in terms of behavior, subjective ratings, and also brain activity. Okay.
So I have a question. So what do you think in your bipolar patients, what would you predict is their signaling deficit, if any?
Well, see, I would say the individual differences are really important here, and it depends. They see kind of inconsistent results, and it also depends on mood phase. So sometimes you see generally it's increased response to reward, and particularly increased willingness to expend effort, that's definitely there. But in terms of brain response, it can kind of go either way, and I think that depends a lot on which mood state they're in.
So you're going to clarify that in your thesis?
Yeah, we'll see.
I'm sure you will. Okay, thank you very much. All right. So, oh, wait. Here. How about this? Isn't this yours? Oh, yeah. What is that? That's a cartoon. Taps his butt. Never gets it. Yeah. I know how that guy feels. Yeah, it's kind of sad. All right. End slideshow. Thank you. So we have two more. And someone's thinking, are they going to be included on the final? I don't know. I'm going to ask the GSIs what they want to do. It's up to them. What's the class think? Yes or no? The class doesn't care. Okay. What? Okay, fine. All right. So listen, we're going to switch gears and get started on a very important topic because we have not a lot to cover. We'll be done before the end of the class. The other thing, the last day of class, I hope some of you will save a few minutes and you can fill out your online evaluations. You know, you should fill them out for me, but I really would prefer if you put the GSIs first because it really matters for them to have evaluations in their dossier as they go forward, for looking professionally to become whatever they're going to do, particularly if they're going to be teaching and be faculty members. So anyway, we'll talk about that later. So language in the brain. Amazing human development. How's it organized? How does it go awry, basically, in disease? And language disorders, there's probably, I'm just going to guess, 500,000 cases of nuance language disorders a year. Again, the brunt from stroke, but there are many other causes for this. So the first point to make really clear to everybody, this is just like neglect is not a sensory problem. Language is a set of symbols that we use as a means of communication. It has nothing to do with the sensory modality in essence. Speech, however, talking, is the mechanical process of language, including articulation and phonation. So I could have nothing wrong with my language network, but I could have a severe cerebral disorder, or my language, my motor would be bad, but the linguistic content of everything would be intact, right? So just keep that in mind, because there are many, many diseases that affect motor structure, and they're not disorders of language. And because it's a set of symbols that we use as a means of communication, you could use any symbol. As you'll see later on in the lecture, people who are deaf use sign language. Right? That's not really something that most people use, but it works quite effectively when you learn it, because you can use it to convey what you want to communicate to the other person. Aphasia is a disturbance of language, the breakdown of the grammar and the syntax, the actual structure of the sentence, the way the words are chosen, the type of words are chosen, the order of the words. Are they real words? Are they made-up words that are not part of the lexicon? Are you connected? Are the semantics of the sentence correct? All these higher-level things are what break down in aphasia. Grammar, syntax, and as I'll point out and show you examples of, what am I doing here? Inability to name and odd words. Inability to name or anomia is the number one most common residual problem from any aphasic disorder. If you're going to be left with problems, it's going to be in naming, which naming is pretty simple. What's this called? Excellent. Okay. And if the patient was aphasic, they wouldn't be able to say a pointer, but if I said use it and gave it to the patient, can you point at the screen? It's the bottom one. Bingo. So an aphasic patient could do that, right? But they couldn't find the word. That's anomia. Thank you. Okay. Now, it happens in any quote modality: speaking, comprehending, vision, reading, sign language, tactile, braille, etc. You can get aphasic disturbances in all these separate input, most sensory, if you will, input modalities to get the information in that you want to turn into some communicative signal. Dysarthria is just poor articulation. I gave you an example in the cerebellar patient. Hypophonia, as we've discussed, is a cardinal symptom of patients with Parkinson's disease. Low volume voice, very common, just like their movements are low. Everything's low, if you will. So these bottom two, dysarthria and hypophonia, they're disturbances of speech, the actual movement of the articulators, but not language. Speech is supported by large parts of the left hemisphere for vocalization. Language is observed by the left hemisphere. This is very important. 98% of right hemisphere males have language in their left hemisphere. This is a very strong lateralization, right? If you think of all the types of lateralization and function you can have in the brain. Women are not quite as lateralized. You'll find a significant percent of women, somewhere in the range of, roughly 7 to 10% of women actually have language in the right hemisphere, not the left hemisphere, or they have a bilateral distribution of language. So that's a very big gender difference. Now, quite importantly, is if you look at left-handers, only left-handers, not right hemisphere. These are genetic left-handers, which means there's lefties in the family. This is different than a pathological left-hander. A pathological left-hander means basically your kid developed language. They were 6 years old and a happy little kid. They fell off their bike and had a massive injury to their left hemisphere with a big something requiring surgery. They will lose their language, just like an adult would lose their language. But if it happens before the age of 8, 9, or 10, the right hemisphere will likely take over the language, right? So in a kid who's had an early onset damage to the left hemisphere, they're lefties because they've been forced to use the right hemisphere. That's different than a genetic left-hander. Your Uncle Tony was a left-hander. Your whatever, okay? So when you look at those numbers, it's roughly a third of left-handers have genetic left-handers have language in the left hemisphere. A third of left-handers have language in the right hemisphere, and actually a large percentage of left hemisphere have bilateral language distribution. So if you put them in an MRI scanner and you do a language task, you'll see bilateral activations, not predominantly left hemisphere activations. These are very important numbers in terms of understanding. You know, for instance, if you get a left-hander in with a left hemisphere stroke, it's probably quite likely they've got over a 50% chance of recovering quite quickly because they got language in the other hemisphere, just that little finding. So these are, and by the way, the kids' recovery is really pretty important, this window, because it tells you that even in this most central human capacity, strongly lateralized, you have tremendous brain plasticity. A whole big function, language, can be taken over by the opposite hemisphere. Some of the takeover of function is actually in non-lesion components of the left hemisphere, but a large part of it is actually the homologous area in the other hemisphere, and now starts working like Broca's area, as you will talk about in more detail, or Wernicke's area. Really amazing ability for plasticity. If we understood it, why the kids can do this, It would be tremendous. It would help us unlock new tools for rehabilitation in people above the age of 10 or people in their 50s or 60s who've had a stroke and have a very bad, let's say, for instance, aphasia. There's always some degree of recovery. It takes a very long time. I saw a patient many years ago, 20, maybe more, who had a gunshot wound to his left hemisphere in World War II. He had his left hemisphere completely gone, basically. He had right hemiparesis and a global aphasia. He had his follow-up records for many years in the Veterans Administration, and he typed every year as a global aphasia. No output of language, no comprehension. And then slowly something happened. I saw him 50 years post, and he really only had residual anomia. But it had taken him 50 years to reorganize, whereas it takes a 7-year-old kid 6 to 12 months. What's going on? We don't know. Please, somebody here, figure it out. That will be great. What you characteristically see in paraphasic patients is, one, problems with naming, right? What is this? You'll see an example. They can't name it, but they know what it is. It's not a visual perceptual problem. They cannot access the lexicon, the word linked to that object. They also make frequent paraphasic errors. These are, para means associated with aphasia. And there's a couple types of errors that you see, and they're all quite interesting. You see many neologistic errors. That means the patient has said something that's not part of the lexicon. Here's two examples. Glipt, crint, they're not words, right? Or they make a very interesting paraphasic error. It's a similar meaning, so they're in a sentence trying to say something and they say knife instead of spoon. Or they could say football instead of basketball. Very interesting, the linguists are very interested in this because it gives them some insight into how your semantic representation is distributed in the brain, which of course is all the hot news if you're interested in computer science and AI and Google deep learning, et cetera, et cetera. These are all taking advantage of massive connections in the language domain or in the visual processing domain, trying to come up with more and more interesting semantic connections of words or phrases or et cetera. So these are less common, but probably the most interesting. Then there's a phonemic paraphasia. That's really simple. It sounds like. So cat for rat. Schoon for spoon, right? It's got the same phonetic structure or very close. You're just a phoning off. And these are all tremendously common in aphasic patients. The neuroanatomy is really important, and this is a study done in 1968 by the great behavioral neurologist Norman Geschwind, and he did a really simple study at the Boston VA. They had 100 brains that they'd been keeping. They didn't really know the handedness of the brains, so there was a big error term in how many were left-handed and right-handed. But even with that, he went ahead and began to measure the size of the temporal lobe, because he knew the temporal lobe was critical in language processing, as you'll see in a minute when we talk about Wernicke's aphasia. He did a super simple study. He basically measured the size of the temporal plane. There's the temporal lobe pulled down. There's primary auditory cortex, and he was particularly interested in this back part of the left temporal lobe. It sits right here. It's your superior temporal plane. And lo and behold, he found when he actually measured the volume of it, it was, now we're going to look down. We're going to do a slice like this, and now we're looking down on the brain. So this is the top of this superior temporal plane. Here's your frontal lobes. Here's your occipital area. Primary auditory cortex, Heschel's gyrus, A1. What he found is in these brains, there was a very significant increase in the size of the left posterior superior temporal plane compared to the right. And he posited that this was one of the biological underpinnings of our hemispheric asymmetry for language. And I think he was correct then, and he still remains correct. There's been, of course, fine-tuning in many of these things. For instance, we know if you look at other species, they're not asymmetric, except if you get to the very highest level of the primate, non-human primates, they show some slight degree of asymmetry in their left and right superior temporal plane. I'm not sure Someone can maybe tell me on Wednesday, I think it's baboons, but it might be gorillas, show a slight degree of asymmetry. Similarly, in the few unfortunate cases of young kids dying in car accidents with dyslexia, when they looked at the superior temporal plane, they actually found abnormalities in the cortical organization of some of the layers of the cortex, suggesting, again, there's some of the thing when you think of dyslexia, there may be something wrong in the brain. There's been subsequent imaging studies that have shown diffusion tensor changes in the superior temporal plane, connectivity of kids with dyslexia, and so on. So this is a remarkable, really simple discovery. You go down to the morgue, you take 100 brains, you measure them, and you change everybody's view of how the brain works. Pretty nice. Pretty nice. We know several other things about the anatomical structure, and I'll stop here and we'll pick this up on Wednesday, but I just want to put it in your mind. If I do an fMRI of language and I have you perceive, I say to you tree, and you say baseball or whatever, we get these areas activated. It's been known for years. Remember we talked about the uncinate fasciculus in terms of biology of emotional regulation, orbital frontal amygdala, amygdala orbital frontal, back and forth, really quick. There's another massive fiber pathway called the arcuate fasciculus. Here it is with a beautiful wet dissection of a real brain, and here it is with diffusion tensor imaging. And when we pick things up on Wednesday, we'll talk a little bit about this being the highway of information flow in your language network. Okay. I have office hours tomorrow, by the way. Thank you. Thank you very much. That was great.